Okay, my friends, as promised, I said everything that you have been taught was wrong. This is another thing. I just saw that Biden has COVID. I'm going to talk about that because they should be given him probiotics, and I'll show you why. What are bacteria? Bacteria, enzymes, and chemicals. What are they? Bacteria are living cells. Bacteria are single-celled organisms. They have a flagella on them. They can scoot all around through your body. They have capabilities of consuming waste of all kinds of different types. They can reproduce. They actually produce enzymes. Better said, bacteria are the factories that produce enzymes. The factories. Well, what are enzymes? They are your health. So don't forget, enzymes are probiotics. They're biotics that are working for you. You've got probiotics and you've got bad biotics. The ones that are bad are the COVID coming in to try to kill you. You need some good ones to fight back. Now, where do you get them from? You get them from probiotics. All right, let's just read this. It's very, very simple. This is Medical News Today. Enzymes, which are these extremely elegant molecules, how they work and what they do. All right, enzymes help speed up chemical reactions in the human body. When they say help speed them up, you know what they're talking about? They're talking about a chemical reaction that could take 100 years. They make happen in under a quarter of a second. That's how incredibly elegant the process is of breaking down and putting pieces back together again. They just don't even understand it yet. They are essential for respiration, digesting food, muscles, nerve functions, synovial sheaths, everything, glucose absorption, every single thing in your body is produced by chemistry, and the enzymes are the only thing that does the chemistry. Otherwise, you're just a bag of chemicals. They're just going to bump into each other. Enzymes are the things that direct their specific biological role, and there's thousands of them, and that keeps you from getting your nerves all shortened out inside, and it keeps you from your skin getting abraded and your lungs getting attacked. And this is what probiotics too. And guess what the National Institutes of Health has to say about them and COVID? Where do you see this? It's unbelievable. Okay, here it is right here. It's probiotics in the prevention and treatment of COVID-19, current perspective and future prospects. This is back from March of 2021, so a little over a year ago. And its conclusion is quite obvious, is that we should be using probiotics. Now, let's go right down to the conclusion, and it says, therefore, supplementation of probiotics in high-risk and severely ill patients, frontline health workers, might limit the infection and flatten the curve. However, there's no random controlled trials. But on the other hand, circumstantial evidence has supported the presumption probiotic supplementation decreases severity of COVID-19, including mortality. And they come back here and they just talk all kinds of good things. Listen to this. Evidence supports probiotics' role in regulating the immune system, and it has for a long time, so that's nothing new. 10 years ago, they had almost the same report as this. Same place, National Institute of Health. All right, so probiotics suggesting a definitive role for probiotics in viral infections, which this is, COVID virus. Probiotic supplementation could reduce the severity of COVID, morbidity, and mortality. Probiotics can inhibit cytokine storms by simultaneously boosting the innate immunities and evading the exaggerated adaptive immunity, which is challenged to respond to viral onslaught like this, because it's a brand new novel disease. It's never been seen before in us, I know of. Now, probiotics induce suppression, which is, they say, probiotics induced suppression of the inflammatory cytokine responses may prevent both the severity and the occurrence of acute respiratory disease syndrome. making probiotics an attractive adjunct, which means add-on. And then it goes on to all this stuff, and then it says, you know, it could transform things around a pandemic, around the globe, and so forth. And it should be supplementation in high risk and severely ill patients, frontline workers might flatten the curve. Everything good. I haven't heard a word about taking probiotics. I don't know if Joe Biden's taking them, but I'd say, Mr. President, get yourself some probiotics. All right. So you know that bacteria are the factories that create these enzymes, and what do? Digestive system. They break down all the food. Enzymes help break down the food from larger complex molecules into ones that, like our glucose and the things that are the little atomic molecules, so that you can reassemble them to whatever you want. Now, they listen to this, DNA replication, and you want to see this actually happen in real time, blow your mind. Each cell in the body contains DNA. Each time a cell divides, the cell needs to copy the DNA. Enzymes help in this process by unwinding the DNA coils. These things are chemists. They know what they're doing. Liver enzymes. Well, you're going to have toxins all throughout your body. And you have to have enzymes to break those toxins down. The liver breaks down toxins in the body. To do this, it uses a range of enzymes to facilitate the process, destroying the toxins. Well, what do you mean destroying the toxins? They blow them up? No. They attach to them and make them attractive to salts or acids or other areas that drain them out of your body. They make them non-toxic, basically, by attaching, I believe it's probably methylation for there too. Let me show you about methylation in CpG islands. This is stuff nobody knows about or is talking about or thinks about, basically. Alright, every time you hear about cancer, oh, the guy's got a bad gene. Well, what does that mean he's got a bad gene? Well, he didn't inherit a good gene, or he got exposed to some kind of toxic chemical that it killed and broke that gene up, and now the gene's defective. And I said, well, let's examine that in a little detail. Let's talk about how do genes work? How do genes work? Well, they turn on or they turn off. Is that correct? They turn on or they turn off. How do they do that? Well, they just do. And they methylate. I said, well, what is methylation? Methylation basically is an on-off switch, and that's basically all it is. It's a chemical bridge that turns on a CpG island gene, right? So they have them DNA methylated CpG islands associated with gene silencing, right? Why would they be silenced? If you don't have the enzyme to turn it on, it's not going to turn on, so it's silenced. Well, what creates the enzyme? We know that the bacteria is the factory that creates the enzyme. And why is that factory dead? Is because of a toxin, yes. But the toxin killed the bacteria. The toxin didn't kill your cell. The toxin killed the bacteria. The bacteria is no longer available to create the enzyme. The enzyme is not there. The DNA methylation of that CpG island is not going to work. This is a great article, by the way. This is by original research article, Epigenetics, Wan Jun Lim, Kwang Kim, Jae Kim, Sungom Jong, and Nashim Kim. And it's very, very good. It's very simple to understand. And here's what it's saying. DNA methylation is an on-off switch, and it plays an essential role in regulating gene expression. It's either on or it's off. CpG islands are DNA methylation regions and promoters known to regulate gene expression, on or off, through transcriptional silencing of the corresponding gene. So they think the gene should be there and working all the time, and there should never be a problem with it unless there's no gene expression. Now, DNA methylation at these CpG islands, we're going to talk about that in a minute, is crucial for gene expression and tissue-specific processes. Sweat glands and hair follicles and skin being able to be shed, and all kinds of things. Every single process in your body is regulated by these genes, but the genes are turned on and off by enzymes. And enzymes are products of the bacteria. And the bacteria live in the interstitium. I've gone over this so many times. Now, at the current time, a limited number of studies have reported on gene expression with DNA methylation in diverse adult tissues at the genome-wide level. So they're really not doing much. Expression levels are rarely affected by DNA methylation in normal adult tissues. However, statistical differences in gene expression level correlated with DNA methylation have recently been revealed. So now I really started looking at it. In this study, we examined 20 pairs of DNA methylomes and transcriptomes from RNA sequence and reduced representation bisulfite sequencing data using adult orgy chicken tissues. Now listen to this. A total of 3,133 CpG islands were identified from these 20 tissue data in a single chicken sample, which could affect downstream genes. What does that mean, downstream genes? Well, there's over 3,000 sites in this little piece they check. And who knows where it is, because it's going to have different DNA and different DNA signatures and different genomic signatures depending on where it is. If it's near an organ, or if it's in the kidney, or if it's in the liver, or wherever it is. So they're just saying they found 3,000 in just this one little sample, wherever it came from. And they could affect downstream genes, yes. Everything is such an involved process. It's called nucleophilic substitution using enzymes. I'll show you that in a second. Nucleophilic substitution happens in aqueous solutions, and we are just nothing more than a bag full of aqueous. All right, so analyzing these CpG islands and gene pairs, statistically correlated, and these are all the things that they say give you cancer. All right, so if there's no expression there, the gene's not working. The gene doesn't work, you get cancer. That's how they're taking it. Okay, I'm just getting ready to do this, and I went to look for this guy's last update on this, and I think maybe it was a couple of years ago, and he just put it out today, July 20th, 2022, I think just a couple of hours ago. Tim Sodenberg, University of Minnesota, Morse. Now, what's he talking about? My favorite subject, nucleophilic substitution reactions. And what does that mean? It means invading groups and leaving groups. And groups of what? Groups of different atoms. And why would they invade and others leave? Because of their affinity for that. It's actually magnetic interaction, because it's dipoles. Now, he'll talk about it, and it was good the last time I saw it, but all I can really care about is to understand this is the process. He's talking about, this is the first time we'll be seeing real biological or organic reaction mechanisms. Nobody's doing this stuff. So he comes down and says, one very important class of nucleophilic substitution reactions in biochemistry are the SN2 reactions, catalyzed. Catalyzed means using enzymes. Catalyzed by this acetylmethionine SAM dependent methyltransferase enzymes. Enzymes. Where did enzymes come from? Do you remember about the factory? They come from bacteria. If you don't have the enzymes, what do you think is going to happen? The guy comes down and he says, hey, go throw a couple of those enzymes over here. He said, I don't have any. He said, what happened? I said, the bacteria that uses that, somebody ate a whole batch of zinc one day, and that thing just can't tolerate that, and they died. He said, well, don't you have anybody else can take over? He said, no. I said, well, what are we going to do? He said, well, I don't know, throw a bunch of stuff at it, and that's called a cytokine storm. All right, and that's down in here somewhere, too. He talks about cytokines. I think it's in here, but they're all over the place. But enzymes, catalysts, transfers, cleavages, breaking bonds. That's what these things do. Hold on a second. All right, I'm just going to read this quickly. I think I've made my point, I hope, or I will. The CPG island sites are regions of DNA where a cytosine nucleotide is followed by a guanine glutide in the linear sequence of bases along the 5' and 3' direction. CPG sites occur with high frequency in genomic regions called CPG islands. That's what we're talking about now. And I did go to I took genomics at Johns Hopkins, so I'm a little bit familiar with this. Now, cytosines, you've heard of cytokine storms. Well, what's a cytosine versus a cytokine? We're going to find out in a second. But you see what these things are? Look at the elegance of that molecule. And that's what it's got to be every time. It's got to be just like that. And that has all kinds of different, actually polar areas, like at the tips and in between the coils, there's going to be some kind of energy. I think that's where they mostly attach, because they can snake down and fall in and lock on and move a little bit because they're flexible. And they attach to whatever they want to kill, and they kill it or break it down. And then they leave. They go to the next guy. That's the beauty of an enzyme. All right, now, so what do we have here? Cytosines in CPG dinucleotides. That's bipolar, di. I mean, it has 2 nucleotides. Can be methylated to form this 5-methyloxine. Enzymes add a methyl group are called DNA methyltransferase. All there is turning things on and off. In mammals, 70 to 80% of these cytosines are methylated. Methylating the cytosine within a gene can change its expression. Turn it on or turns it off. A mechanism that is part of the larger field of science studying gene regulation called epigenetics. Methylated cytosines often mutate into thiamines. Well, all it is turning something on and off with an enzyme. But you don't have the enzyme, you can't do the job. You don't have the enzyme, you don't have the bacteria. You don't have the bacteria, you're in trouble. All right? There's more than just the bacteria, obviously. You have to have a good, functioning, working, ready to go gene, but it has to be turned on or off. It's just like the guy doesn't turn a switch on, then nothing happens. Okay, from everything I could figure, these are magnetotactic bacteria. And they're widespread, they're motile, diverse prokaryotes, biomineralized, which means they got a lot of different minerals in them, and it's biological. Unique organelle called the magnetosome. These consist of nano-sized crystals, I mean they're almost nothing, of magnetic iron mineral that is enveloped by a lipid bilayer membrane. So they have a tiny little envelope that these things go in, and then they go out and attach and kill or break down things. That's the way I'm taking it. Magnetotactic bacteria. All right, we've been talking about cytokines, which is like a cytokine storm, attacks your whole body. And they're made of proteins, of small regulatory proteins that are actually enzymes that regulate the cells of the immune system. So you can attack your own body with your own immune system if you don't have the right stuff to fight back in the first place. It'll just go get any troops it can get, and they'll come down and they'll kill you because they're trying to fight something they have no ability to fight. Now, what is a cytosine? It's a different type of a chemistry, that's all it is. It pairs with guanine and DNA and RNA. It's got a base of this, and then, I don't know, things attached to it. I'm not really sure, to be perfectly honest with you. But they are 2 discrete, separate things, but I think they're very closely related. Okay, I think I've pretty well established that it's enzymes that turn on or off the genes, and that is impacted by methylation and the actual physical property of the DNA. If it's turned on, switched on, or turned off. If it's off, you're not going to get the product that you need to break something down. It's as simple as that. The gene might be there, but it won't express. And they say, well, it doesn't express, it must be defective. No, it doesn't express because it's not being told to turn on by this CpG methylation. You got to turn it on.